All right. We are live. So, hello everyone, and welcome to the 2022 Fall Headwaters Science Institute Research Experience Presentation Night. My name is Daniel Dudek, and I will be your host this evening. I'm the manager of this program and mentor to all of the students here tonight. Before we get started, I want to give you a breakdown of the program so that everyone understands what we are highlighting. And also, I have a few announcements. So each of the students presenting here tonight has spent the last 10 weeks designing their own research project from scratch based on their own curiosities. We have helped them hone their study design, analyze their data, write a manuscript, and create a presentation that all of you will listen to soon. This is truly the summation of the last 10 weeks of work on top of their typical school workload. So they've devoted a lot of time and effort, and you'll hear about some of the highlights and the struggles that each of them has gone through throughout the program. Now for a few quick announcements. We are still accepting enrollments in the spring program that begins late January. So if you find this program as exciting as we do, please share it with any parents or students who may be interested. Second, we are accepting donations for the Give Back Tahoe Giving Tuesday competition. If you can, every penny helps bring unique programs like this to the masses. All of that being said, let's get started tonight. So after each presentation, we'll have some time to take questions from the audience. So please submit any questions you may have in the comment section on Facebook Live or YouTube Live, and we'll take one or two. So for our first presentation tonight, I would like to introduce Rushika, who's going to tell us more about her algae research. So I'll bring her up now. Give me 1 second. All right, Rushika, how are you doing? Are you ready?
Yeah, I'm doing good.
All right, what an exciting time. All right, so with that being said, I'll let you take it away.
Hi, my name is Ruchika, and my presentation is on the relationship between climatic factors and freshwater microalgae species. So, to introduce microalgae is a unicellular phytoplankton that is found in both seawater and freshwater. However, this presentation will focus more on the freshwater species. They are essential primary producers and are able to photosynthesize, which allows them to produce oxygen that is essential for living organisms. Many are also used in medicinal or food products. However, others may be toxic and lead to death in many organisms. They are also used to aid in the production of different biofuels. So these are the three freshwater microalgae species that my research is based on. So to start off, spirulina. Spirulina is a blue-green microalgae that has a high nutritional value, containing an abundance of vitamins and proteins. So this is one of the microalgae that would be used in medicine or vitamins that I talked about earlier. Chlorella. Chlorella is a green algae, and similar to spirulina, it has a high nutritional value that has been shown to improve total cholesterol levels, low-density lipoprotein cholesterol levels, systolic blood pressure, and diastolic blood pressure. Microcystis. So microcystis is a blue-green algae like spirulina. However, it produces the toxin microcystin that has been shown to cause death in both humans and marine organisms. So it travels to the liver and disrupts normal cellular activity by inhibiting protein photophases, which can be lethal to these organisms. So there are many differences between the climates and environments that these different species of microalgae are able to survive in. These differences and understanding the suitable environments that the algae are able to live in can help us gain a better understanding of how climate change may affect these important primary producers. So a study was actually done of the microalgae Dinoliella at the coastal range of the Atacama Desert. It showed that they were actually able to adapt to the dryness and heat of the desert by forming biofilms on the spider webs. and they use the water condensation on these webs as a source of nutrients. So this is just one of the many examples of events where microalgae were able to adapt to their environment, and analyzing the difference in the different species ability to grow under these different climate conditions can be the first step in understanding the versatility and adaptability. So this presentation will look at the climatic differences between the freshwater microalgae spirulina, microcystis, and chlorella, and compare the different factors under which they are able to survive under. So my research question was, what are the climate differences between different species of freshwater microalgae? My independent variable was the type of microalgae, which was spirulina, microcystis, and chlorella, and my dependent variable were the bioclimatic factors, such as precipitation and temperature. My hypothesis was that the blue-green algae, spirulina and microcystis, will prefer the higher temperatures and more rainfall, while the chlorella would prefer lower temperatures and less rainfall. So my methods were that I obtained location data for each of these microalgae species, chlorella, spirulina, and microcystis, from GBIF, which is an open access biodiversity database, by using Wallace, which is an app for ecological modeling of the different species distributions. And then I used R to pull the bioclimatic data from each location. After that, I explored the existence of the differences between the different species of microalgae for each of the climatic variables by visualizing the data in Data Classroom. And after that, I ran statistical tests such as the post hoc Tukey's range test, linear regression, and ANOVA test to find if there was any significant relationship between each of the variables. So these are my results. The first graph represents the relationship between the mean temperature of the driest quarter and the mean temperature of the warmest quarter. So as you can see, there was a relationship between the mean temperature of the driest quarter and the warmest quarter, as the p-value was lower than 0.01, and the r value, or the r squared value, was 0.596, which shows there was a significant relationship. And as you can see, the microcystis was more prominent in areas that were higher in temperature for the warmest quarter and driest quarter. And in this graph, it shows the relationship between the annual precipitation and the genus. After running the ANOVA test on this graph, the overall p-value was also under 0.01. But after running a post hoc Tukey's test, the p-value of the spirulina and microcystis was 0.81, which does not show any evidence of a difference. However, the chlorella was significantly different than the other two. This graph shows the relationship between the genus and the annual mean temperature. After running the ANOVA test, the overall p-value was 0.11, which shows somewhat of a difference between the groups. However, the p-value for the spirulina and microcystis was 0.99, which shows that the groups were not different. So through comparing the mean value of the temperature of the warmest quarter and the mean value of the driest quarter, we can see that through the p-value of less than 0.01, there's a very significant relationship between the two, which can be explained in that the environment gets warmer as it also gets drier. And the microcystis seem to survive in these warmer conditions better than the spirulina and chlorella. And in graph number 2, through comparing the variability of the annual precipitation of each of the microalgae species, the chlorella is clearly surviving in climate conditions with much less precipitation, better than both the microcystis and spirulina, and supported by the post hoc Tukey's range test. The microcystis and spirulina have a higher p of 0.81, which does not indicate a difference between them. So this indicates that the chlorella is just significantly different than both of the two species, as it is more suitable in a habitat with a lower annual precipitation than the spirulina and microcystis, which are more suitable in a habitat with a higher precipitation. In graph 3, it compared the annual temperatures for the different microalgae species. The p-value was 0.11, so there was somewhat of a difference between the group. However, since the p-values for the spirulina and microcystis was 0.99, this does not show a difference between the two. However, there was a slight difference between the chlorella and the spirulina and microcystis. So overall, the spirulina and microcystis had the highest p-values when compared to each other. This shows that there was little evidence that they were different from each other, and it supports my previous hypothesis that the blue-green algae would have similar suitable habitats since they are the same type of algae. They may prefer warmer temperatures as the algae may be more able to move through the water easier and float to the surface faster, which is essential for their process of photosynthesis. Since chlorella, which is a green algae, is a protist and differs from the two other species, which are bacteria and blue-green algae, this may play a part in its growth in areas with lower annual precipitation. So the raindrops may disrupt the algae on the surface and affects its photosynthesis for the chlorella. So this research helps us understand the differences between the suitable habitats of these microalgae species, and it can be really important to know how climate change may affect these algae, and we have to try to stop this from happening. So a source of error could be that since this database was mostly based on scientifically grown microalgae and is an open portal, this may not represent the entirety of the species population, but it can be still good to know. So future research. As the climate in different ecosystems, including those of microalgae, change, it is important to understand the climatic differences between these species to make an effort to save them. Research into how climate change would affect different species of microalgae or how we can improve their adaptability to changes in their habitat can prove helpful in this goal. And this is my work site. Thank you for listening.
All right, great job, Ruchika. Fantastic job. So as we wait to see if there are any comments that roll in, the first thing that I would like to ask you is what is one of the highlights for your project? Throughout this whole experience, can you list one of the highlights?
Well, one of the highlights would just be the entire project. I feel like it was great to explore microalgae because it was something I've always been interested in, and to gain a deeper understanding of the climatic differences and the locations between them was really cool.
Yeah, and then I guess for the struggle for your entire process, your participation in the program, tell us a little bit about the struggles you had, because I know it's quite a story.
Yeah. So originally I was planning to do the experiment. It was growing microalgae. However, the materials were extremely late, so that did not work. And this was kind of like a plan B and very last minute, but I'm happy that it still worked out. And yeah.
Yeah, that was one of the detriments. It's always a detriment whenever students are trying to create a project around growing, whether it's plants, algae, or any of these types of things. There are just variables out of your control. You had no control whether or not the materials were going to be shipped to you. But yes, I'm very glad that you were able to pivot and create an amazing research project on studying the ecological differences or the climatic differences between these different microalgae. So again, great job, fantastic effort. All right, Rushka.
Thank you.
Yep. All right, so next I would like to introduce Elise, who is going to tell us more about invasive and native plant species in an area close to her. So again, this project is going to be, related to an area very close to her, which is oftentimes a great topic in these programs. Some of the students really want to investigate something that's, in their backyard instead of doing more of a data mining project like we saw with Ruchika, and then later on we'll see another data mining project. But projects like this, students are able to actually get outside and collect field data themselves. So that being said, I will let Elise take it away.
All right. Hi, I'm Elise. I'll be presenting on my project, which is the relations of land development and its effects on native and invasive plant coverage and its diversity. To start off, the Sacramento-San Joaquin River Delta, also known as the California River Delta, is an extensive network of around 1,000 square miles, which consists of freshwater and freshwater marshes, as well as brackish water. It has been heavily developed for agricultural use due to it being very fertile land from the sediment deposits. Levees have been created and replaced much of the natural bank that the delta would normally have in order to make farmland and residential areas. Invasive species in the delta. Many species of introduced plants have negatively impacted recreational activities. In the top right corner, you can see that there's lots of water hyacinth clogging one of the waterways, preventing boats from getting past. Plants such as water hyacinth, alligator weed, and sponge plant can push out native species that would otherwise occupy that area. The majority of aquatic plant species are introduced accidentally from people who grow it for ponds and release it, or dump from ship water ballast. Terrestrial species can also accidentally be tracked in or planted for landscaping purposes, and then they end up getting into the environment. Some of the biggest plants that are an issue in the delta are the common water hyacinth, which is a very buoyant plant. It can float long distances and create very thick mats, and they also prevent submerged plants from photosynthesizing. Another one is alligator weed, which is a terrestrial or immersed plant that grows very big mats of horizontal, foot-high plant structures, and it takes up space that native plants would usually occupy. Another one is sponge plant, which is a floating plant that has a very high propagation rate and covers very, very vast areas of mats, which prevent submerged plants from photosynthesizing, as well as preventing the water surface from being disturbed, making it so it cannot be oxygenated. For my research project, I wanted to figure out if there was actually an impact on native plants from agricultural development, and if invasive species impact native plants from their introduction and proliferation. These two pictures are sample sites that I visited. The bottom left-hand corner is the Dutch Slough, and there's a large marina located right next to it. The top right-hand corner is the Consumnes River Preserve, which is one of the less developed sites of all the areas visited, and while technically not necessarily part of the main delta area, it is still connected with waterways. My overall research question was, how has agricultural development affected coverage between invasive and native species in the California Delta? My independent variables for this research question were the location and the terrain type. My dependent variables were plant type coverage and species diversity. For my hypothesis, I thought that it was likely that invasive species will have a higher coverage in areas where agricultural development is more prominent, and native species will do better in areas where development is less prominent. In order to take these samples, I launched a boat from the Tiki Lagoon and Marina boat launch. From there, I headed up northwest and took samples that were a 10 by 10 foot section with 50 square feet being immersed vegetation and 50 square feet of terrestrial vegetation. For other locations that I visited, I took samples along the main section of the Sacramento River and the Consumnes River Preserve. For the overall results, this graph here shows the invasive abundance compared to native abundance. Overall, abundance can only equal 100% due to the area coverage, and missing percentages account for water and bare ground, which did not have any foliage covering it. It also shows that there's a limited amount of space that plants can occupy. This graph here shows the invasive species count, individual species basically. As you can see, this graph has a relatively similar mean value for both the agricultural and less developed areas. There is a p-value of 0.991 for this data, which means it is very statistically insignificant, and location type has no impact on invasive diversity. This graph here shows the native diversity compared to the location type. As you can see, there is a mean value that is much higher in the less developed area, being about 3.5, while the agricultural area has a mean value of about 1.5, showing that there is a much higher mean value in the less developed area and therefore higher diversity. This graph has a p-value of 0.03 for this data, meaning it is statistically significant and that the location type does indeed impact native diversity. These two graphs here, figure 4 shows native diversity percentage coverage. It has a p-value of 0.17, meaning there may be some correlation between the location type and the percent coverage of the area. It has a higher mean value in the less developed area and a lower mean value in the agricultural area. Figure 5, the one on the right-hand side, shows the invasive percent coverage compared with its location type. It has a p-value of 0.37, which indicates that there is little to no evidence suggesting that location type has an impact on an invasive species percent coverage. However, the graph does show that there may be some correlation. This graph here, figure 6, shows the native coverage of the area compared with its diversity. There is a very clear increase in coverage as the diversity goes up. The p-value is less than 0.01, which shows that there is a very strong correlation between the two categories. As native diversity goes up, native abundance also goes up. This is also a good argument for the idea that biodiversity is important for the ecosystem health. This graph here shows invasive diversity compared with invasive percent coverage. The line of regression, which is the green line on the graph, shows that there is a correlation in increase in species and as the increase in coverage as the diversity increases. However, this may be due to the two extreme values on the right-hand most side of the graph. Also, only 50% of the abundance is explained by its diversity, as shown by the R-squared value of 0.505. For the conclusion drawn from my data, it shows that agricultural areas have slightly less native plant diversity and coverage overall. The invasive plants are present in larger quantities and have more diversity in agricultural areas. And native plants have less presence in agricultural areas, like do the invasive plants taking up space and resources. The overall conclusion is that agricultural development, in conjunction with invasive species, have negatively impacted native plant diversity and coverage in the Delta region. Some things that would probably improve my data would be if the boat samples were taken much closer to the shore, as the boat might have been beached if it was taken very close. Also, some species may not have been documented due to them being too small or not seen. Also, time constraints, because some of the areas weren't super accessible by car. It took a long time to get to some of the sampling locations. Additionally, some of the locations that were samples were done out of accessibility, since some of the areas were were very swampy or had plants that prevented me from accessing them, and they're also mostly chosen at random. Overall, to improve the data, a larger sample size would be taken. Likely there would be more diversity in the types of sample locations, with better precision, and also data taken over time would be ideal, as some species do not show up during certain seasons. For future research, based on what I've done for this project, I would like to probably do a terrain-based plant type coverage investigation, looking at to see if invasive species are better at covering terrestrial areas as opposed to aquatic areas, or vice versa. Also, I would like to do a similar investigation to this one, but it would be focused on smaller local waterways such as canals and small creeks. Also, a species comparison on an abundance and well-being of fish species, both invasive and native, would also be interesting to do. This is my work cited. Thank you for listening.
All right. Great job, Elise. Again, fantastic job. I know your project, there was a lot of field work associated with your project. So yeah, to start off, well, why don't you talk to us a little bit about, your field work efforts?
So basically, the initial sample taking, I had to go out in a boat, which was slightly difficult since the boat wasn't like a kayak. We had to drive it so it wouldn't get caught in all of the submerged aquatic plants, what do you call it? Water milfoil was one of the issues, and it would get caught in the rotor of the boat if we did get too close, if it was on. And that actually isn't an example of an invasive species impacting recreational activities in the Delta. And also, I didn't exactly have like waders or anything to go tromping through protected areas, so I couldn't get samples in sections that were very marshy. So mostly sticking to trails that were already there, or areas you could pull off on the side of the road that were right next to the Delta, or going to parks that had the waterways connected to it.
All right. Yeah. So that sounds like a bit of a struggle was actually doing your field work. But again, great job collecting your data. But can you tell us a highlight of your entire experience?
Well, I've always liked looking at aquatic plants. This project has been very entertaining, especially the Delta expeditions, going out and just looking at all the different interesting types of plants and kind of what they've done to the Delta region. It's been very, very fascinating. You can kind of see how the plants have affected all of the waterways and stuff. It's kind of been a little bit clogged in areas, and it's kind of interesting to see how the invasive species have changed how the Delta works, as well as the human impact on the area. You could see all the levees and stuff, or they're just full of rocks, or like intake valves or agricultural land right on the other side. And the water levels are vastly different from where the developed land actually is.
Mm-hmm. All right. Well, again, thank you for, showing us and talking to us about your research. Again, fantastic job. All the efforts and struggles you had throughout the process definitely paid off. All right, everyone. So for our third presentation, I would like to introduce Shreya, who is going to talk to us about water quality in her local area. Okay, so let me bring her up. Shreya, how are you doing today?
I'm good, thank you.
All right, are you ready?
Yeah.
All right, with that being said, I'm going to...
Okay. Hi, my name is Shreya, and today my presentation is about the effects of polluted water compared to the non-polluted water in the Bay Area. So, wastewater pollution is one of the most common sources of pollution to the food chain. It is mostly deathly and contains toxic compounds that damage the health of aquatic animals and those who consume them. Water pollution is not just caused by littering and industrial waste. It commonly originates from oil leaks, marine dumping, wastewater, land runoff, and global warming. Drinkable and non-polluted water quality can be assessed using physiochemical and biological factors, and tested by testing potential hydrogen, pH, and total dissolved solids, TDS. The pH determines how acidic the water is by measuring the relative amount of free hydrogen and hydroxide ions in the water. TDS measures the amount of organic and inorganic compounds in anything dissolved in the water that is not an H2O molecule. One category of chemical pollution brought on by carbon pollutants is organic pollution. Compounds that are chemically active and present in both organic and inorganic substances contribute to organic pollution. Organic and inorganic pollutants in the water have a negative effect on the ecosystem and people's health. As a result, drinking poor water quality results in individuals being exposed to non-preventable health risks. As has been extensively documented, residents can use the pH and TDS as a quick and accurate instrument to evaluate the quality of water. The main objective of this study is to evaluate the water quality and present the use of the pH and TDS as a possible monitoring tool for the drinking water quality of the marine life and public network in the Bay Area. My research is based on a study that has shown that, as of 2021, Foster City's beaches have reached California's top 10 most polluted water sources identified in the Bay Area and have been detected to have harmful contaminants. So my research question is, how does the use of a pH and TDS meter evaluate the polluted water compared to the non-polluted water in the Bay Area, and what are the effects? So my independent variable is the locations of my experiments and location categories, so the polluted versus non-polluted water sources. And my dependent variable is the pollution in the lake detected by the pH and TDS meters. My hypothesis is I believe that the outcome of my research would result in less living organisms in the polluted water compared to the fresh water due to the acidity and all the harmful compounds. Materials and methods. Water pollution is hard to detect solely based on looks, as there are many factors that go into the cause of water pollution, such as bacteria, toxic waste, global warming, sewage and wastewater, and other kinds of chemical and microbiological pollutants. The most efficient way to detect pollution in the water is by detecting the acidity of the water or the nutrients and compounds that the water contains. So this is when I was collecting the pH. So one way to detect if the water is polluted is by collecting the pH of the water. When there is heavily polluted water, the acidity of the water drops or is raised extremely. So a pH meter is what I use to determine the pH, examines the hydrogen ion activity in water-based solutions in order to determine whether a solution is acidic or alkaline, and expresses the information as pH. So I tested 2 polluted sources and 1 freshwater source in the Bay Area. When testing the water, if the pH of the water comes between 6.5 and 9, the water's acidity is less acidic, resulting in it to be unpolluted and safe. However, if the water is tested to be under 6.5 or over 9, the water's acidity is extremely high or extremely low, proving that the water is polluted and unsafe due to the acidity level. So this is when I was collecting the TDS. In water, the more organic or inorganic compounds there are, the more polluted and contaminated the water is. So TDS, the total dissolved solids, is a measure of anything dissolved in water that isn't an H2O molecule. TDS is the quantity of organic and inorganic components, such as metals, minerals, salts, and ions, which are dissolved in a specific volume of water, which I tested at 2 polluted sources and 1 freshwater source. A clean and healthy freshwater score lies between 50 and 250 parts per million, and anything above 1,000 parts per million is unsafe for consumption. The higher the TDS score, the more amounts of compounds are present in the water, resulting in unsafe water. So this is my first graph, and Quarry Lake, which was my freshwater source, which is purple, shows that there's an 8.5 pH, and my polluted sources, which are at Marlin Park and Erkenbach Park, are yellow and green, which are slightly below 6.5 pH. And in this, as you can see, for Quarry Lake, my freshwater source, the mean of the pH is 8.49, which is slightly below 8.5. And for my polluted sources, they have a pH of 6.3. So for my TDS at Quarry Lake, my freshwater source, they have a TDS under 1,000 parts per million, while my polluted sources, Marlin Park and Erkenbach Park, have a TDS slightly above 4,000 parts per million. And here it shows that my freshwater source has a TDS for 279 parts per million, while my polluted sources have a TDS above 4,000 parts per million. So If a pH is less than 6.5, it is considered too acidic, but if it's above 8.5, it's too alkaline. So my pH between my polluted and non-polluted sources show a difference for about 2 pH levels. My non-polluted water's pH is alkaline and basic and about 8.49 as shown in the graphs before, and not drinkable, but it is safe. And my polluted water pH is high in acidity and it's about 6.3, which is not drinkable, it's polluted and almost too acidic for the human body. So for TDS, between 50 and 150 parts per million is considered suitable for human consumption, and any TDS under 1,000 parts per million has no harmful / inorganic contaminants. So between my polluted and non-polluted TDS, it shows a difference of 3,700 parts per million. So my non-polluted water's TDS is not harmful, and it has about 279 parts per million, while my polluted water's TDS has about 4,100 parts per million, and it is considered very harmful and polluted. So this reveals that the polluted water is not only unsafe because of the pollutants, but a large factor contributes to the high acidity of the water and the large pollution of harmful organic and inorganic compounds. So for my polluted water, some factors that contributed to the high water acidity or high pH was the tree roots, rock formations, soil microbes, industrial pollution, chemical dumps, and large numbers of chemicals and pollutants. And when I did visit the polluted water source, there was no marine life that was found. The water was a darkish green color, and there was trash all alongside the shore. While the non-polluted water, which was Quarry Lake, which is a wetland, which has natural resources that filter out the pollutants. The water when I visited was clear, and there was different fish, ducks, crabs, and sea floor marine animals swimming around. So this shows the difference of the living organisms present in both water sources and how dangerous polluted water can be to life. The pH and TDS prove my hypothesis and show a significant difference when comparing the two water sources. So for my further research, I'll research more about living organisms living in polluted water and their immunity, and about the specific organisms that can live in both waters and also more about their immunity, and about the effect of this water on plants and their difference in their growth. Thank you for listening.
All right. Fantastic job, Shreya. Wonderful presentation. Yeah. So my core question is the first question being, have you done a lot of field work in the past, and what was, conducting field work like for you during this experience?
This is actually my first time doing any type of field work on my own time. And I enjoyed it a lot. It was really nice visiting the lakes and water sources. I really liked experimenting with pH and TDS, and I learned a lot more from this experiment.
So would you say conducting field work was a highlight of the whole process?
It actually was. It was my highlight for this process.
Ah, fantastic. Well, what about the struggle? Did you have any struggles?
I had one struggle. One of my water sources was pretty far away, which I had to take a few rounds, but it was worth it at the end of the day.
All right. And I think my final question would be, what's the take-home message to, I guess, the general audience or the people that live around sort of these bodies of water?
I would say to keep water pollution to a minimum, because water pollution has become a big thing in pollution in general, not just air pollution, but it also contributes to water pollution. So, maybe just throw your trash in the trash can and not just on the floor.
Fantastic answer. Fantastic answer. Well, again, great job, Shreya. It was a pleasure being your mentor.
Thank you.
All right. Let me bring you down. And then lastly, Max is going to talk or give us some insight into how air quality influences COVID. So cases and deaths, but he'll get into that. So Max, how are you doing tonight?
I'm doing good. How are you?
Great. So are you excited to, share what you found with everyone?
Yeah. I'm excited.
All right. Well, with that being said, I'll let you go.
All right, so hey everyone, I'm Max, and this is my presentation on the effect of air quality on the coronavirus cases and deaths. So just a brief introduction. So the coronavirus, otherwise known as SARS-CoV-2, is one of the largest threats to the globe right now. It's become a widespread pandemic that originated from Wuhan, China at around January 10th, 2020. It spread across the world, hitting all countries, causing hospitalizations and deaths. And the effects of the pandemic have been uneven, as rates of death have been much higher between countries and even in between states and provinces. The differences that cause varying death rates and case rates has been heavily studied. For example, research has been done previously on the effects of even varying temperatures on the transmission of the coronavirus. This paper is similar in the fact that it's going to examine one difference between different regions that might cause these different case and death rates. So this paper is going to focus on the differences between the air quality of these regions. Different areas tend to have different air quality indices, or the AQI, which I'm going to use to measure the air quality index, which is a value calculated by the particle matter. It includes carbon monoxide, ozone, and tons of other compounds. This paper aims to examine the relationship between the AQI values of states and provinces in the United States, India, as well as Asia, with the worst AQI ratings to determine its effects on infection rates of COVID. So here's my research question. So my general question was just, what effects does AQI have on the rates of SARS-CoV-2 cases and deaths in different regions across the world? So research in the past has indicated that in China, worse air pollution was associated with a higher death rate during the SARS outbreak. And so using previous knowledge, it should probably suffice to say that COVID should follow the same transmission rates and models. So the independent variable for my research was the air quality indices of different states and regions across the world. And the dependent variable, well, there were two dependent variables. The first one is the case rate, and the second one is the death rate of coronavirus in different states and regions. And you'll see the plots. So my hypothesis was, I believe that a worse AQI would result in a higher death rate and higher case rate. And the reasoning is because I believe that in regions where there's high AQI values, people tend to breathe worse air with its higher pollution levels and thus have weaker lungs. This probably means, as in the past has shown, that it will lead to higher case rates and death rates, more likely for you to catch the virus and more likely for you, and the mortality rate must be higher. Okay, and the materials and methods. So the AQI data that I obtained for the United States and India was drawn from Plume Labs, which is above. And for the AQI and COVID case death rates, and the other one for Asia, data was collected from IQ Air. The average AQI over the last year was recorded for major cities within different states and then plotted as the independent variable. And to match the COVID data, the case and death rates for the country or state in which the city belonged in was recorded as well. This data was drawn from the New York Times' coronavirus case count tracker, as well as the Johns Hopkins University's CSSE database. So there's a lot of data mining, obviously, in this project. It was mainly based on data mining, and I didn't actually have to go outside and do any field work. But overall, I was able to draw 65 different data points between a region, its AQI, and then on top of that, the case and then the death rates. All of this was inputted into a spreadsheet and then plotted to view the correlation. So here's the first figure. This is a graph of the case rate versus the AQI values for the three regions, and they're colored differently with different lines. And the best fit lines for all three indicate a negative relationship between AQI and case rate. So here the p-value for this entire group was less than 0.01, which indicates that there's a significant relationship for the entire group. And then looking at the specific regions, for Asia, the R squared value was 0.143 or 14.3%. Americas was about actually 0.0109, and for India, it was 0.193. And so R squared explains the percentage of the case rate variability that is explained by AQI. And this indicates that the best sort of relationship we have is in India, and the Americas is the worst because all AQI values are all clustered around 20, as shown. All the yellow data points are around 20 on the x-axis, which means that there wasn't much spread to draw a conclusion from. And here's the second figure and the second result of my research. And here it shows the death rate versus the AQIs. So the best fit line also indicates a positive relationship between AQI and death rate, although that's actually not true for the Americas region. So this shows that as the AQI worsens, the actual mortality rate or the death rate of coronavirus actually increases. The p-value for this entire group was about 0.02, which means that there's still a very strong confidence that there is a relationship for the entire group. The r-squared value, here Asia was at 0.575, which is pretty good. Americas was once again extremely low at 0.00681, and India was 0.085. Here again, Americas is again the worst, once again, likely because the all Americas data is clustered around at 20 AQI. And it's important to note that the US is actually negative, and I'll try to explain this later on. So for the discussion, one of the results or one of the reasons why I believe that a higher AQI results in a lower case rate is people who live in areas of high AQI and high pollution are probably less likely to go outside, and this actually lowers the chance of contact and transmission, which is one of the reasons as to why you actually see a lower case rate in the areas with higher AQI. However, for the higher death rate, this is probably explained by the fact that in areas of high AQI, people's lungs are weaker because they breathe in worse air. So this means that once you do get coronavirus, you're more likely to pass away because your lungs are already weaker. As shown, the deadliness of COVID increases as AQI worsens. And so there are a lot of errors with data mining. As mentioned previously, for the death rate, the US was actually negative. And one of the reasons for this is probably because of different healthcare coverages and access and all that type of stuff. And also because all of our AQI data is correlated around 20, which is good for us because it means our air is good, but also bad for the data mining project. For future research should be done to actually try to isolate the variables. Between each region, there are tons of minute differences that can cause the variability that I discovered in my research. For example, as mentioned above, variables in healthcare access, coverage, and quality can affect death rates. And that's why the US might have even seen a lower death rate despite a higher AQI, contrary to India and Asia. There are also a multitude of elements such as population density, climate, travel rate, and virus strain that can alter the data for case rates. The results are also quite confusing, and there are a multitude of explanations that are possible, so future research should be done to try to isolate a reason as to exactly why this phenomenon happens. Obviously, and for data mining projects, the larger the data set, the better the results going to be. So although I use 65 points, the research could be much better and draw more conclusive results if you use more and more points. So getting more data points is obviously extremely good in the future. So here's my work cited. And yeah, thank you for listening.
All right, great job, Max. Fantastic presentation, and I think you're selling yourself short. You know, your data mining project was quite a lot. You know, collecting all of those data points from various databases, and then actually uniting all of that data in a way that it's able to be analyzed. You know, I think you're being a bit modest. You know, you did a lot of work there. So again, fantastic job on your project. Extremely happy to see, you know, everything come together the way that it did. So my first question to you is actually it relates a lot to your project. So for the Americas, if AQI isn't really influencing the case or death rate, you know, what's one reason why you think there is variability in case and death rate for the Americas if AQI isn't it?
So in the Americas, probably there are differences in like state enforcement and law enforcement for like contacts and stuff like that. And obviously in different regions and states, there's different quality like healthcare. So like that was one of the things that I mentioned before, like the death rate actually being lower in areas of higher AQI could indicate that if you're in an area of higher AQI, you could probably be in a city, and in cities, like you probably have better access to healthcare. So there are lots of like these differences. And the Americas is kind of like an outlier. It's different from the other two regions that I studied, and it kind of shows that like there's still lots of research to be done to find out like what actually is causing these differences.
Well, great answer. And I guess to match everyone else, what's one highlight and one struggle with the overall process?
So one highlight would probably be like when I actually plot all the data points and I saw the lines, and especially for the case rate one, when I saw the p-value was less than 0.01, it was kind of cool to see that like there actually might be something there that I'm researching, and that like my hypothesis might be correct or it might be wrong, but at least like I proved something. And one struggle, probably the largest struggle, was actually getting started and finding the AQI values. There were a lot of meetings that happened between us where we had to try to figure out like where we were drawing the data from, like what database we should get it from. So it took a while for us to get started, but I think like once I did, it got a lot easier, and then at the end it was quite rewarding to see.
Yeah. Well, again, great job. I know it was a lot of work mining all of the data, but you did great bringing it all together. All right.
Thank you.
All right. So everyone, there's a few more things that I want to say. Definitely, I'm so proud of all of the students who participated in our program. As you saw, they created fantastic projects, and again, these projects were all stemmed from their own curiosities. We helped them take their curiosities, help them design a project, conduct a methodology, and ultimately analyze and present what you're seeing today. So they all did a fantastic job, and they should all have a round of applause. I know they're in the background, probably, very happy with all of their accomplishments. But yes. So hopefully, if you're listening, friends and family of each of the participants, you should definitely be proud of what they were able to do during the past 10 weeks. Yeah. So fantastic job. And lastly, again, we are just so proud of the Headwaters, Science Institute community, and especially since it's Giving Tuesday and we've been collecting donations all day. I believe we actually hit our goal. So, fantastic job. Thank you so much to the community for your generous contributions at this time of the year. Because of you, we're able to continue to provide hands-on science to students of all backgrounds and means. So your donations really change lives and hopefully inspire the next generation of scientists. And we are so grateful for your support. So that being said, have a great night, everyone. And I hope you enjoyed the presentations. Take care.